Hi everyone, welcome back to Neurobiology at Providence College. I'm Joe DeGiorgis. After spending a wonderful year working with Bruce on kinesin at Harvard Medical School, I went to Brown University to do my PhD with Elaine Baer. And in Elaine's lab, I spent my time working on myosins in the squid giant axon. So there are three different types of motor proteins. There are the kinesins, which of course was discovered by Ron and his colleagues. There are the dyneins, and dyneins and kinesins operate on microtubules. And the third group of motor proteins are the myosins, which act on actin filaments, like myosin and actin in muscle. The myosin story in the axon is a very interesting one, but I'm going to skip over that for now because I want to continue with our kinesin 3 story. After I graduated with my PhD, I moved on to work with Tom Rees at the National Institutes of Health for my postdoc. I spent some time in Washington at the NIH, but most of my time was spent in Tom's lab at the Marine Biological Laboratory in Woods Hole, working once again on squid and the kinesin story. The paper we're going to talk about today is titled Kinesin 3 is an organelle motor in the squid giant axon. So kinesin 3 is different than the kinesin that Ron and his colleagues purified. They call that a kinesin 1 now, or they sometimes refer to it as conventional kinesin as the founding member of the kinesin motor family. This publication was published by myself, by Tatiana Petikova, Teresa Evans, and Tom Rees. It was published in Traffic in 2008. Before we get into the publication, I need to talk a little bit about some sequence work that I did prior to this publication. We all know the central dogma. DNA gives rise to RNA, and the RNA gives rise to the protein. And the process of generating RNA from DNA is transcription. And the process of generating proteins from RNA is, of course, translation. And we also talked extensively about Ron and his colleagues and their work to purify the protein. They purified kinesin, the protein, from squid optic lobes, essentially the brain of the squid. And at that time, sequencing DNA and RNA molecules was really in its infancy. So Ron really had no choice other than to go after the protein, that is, biochemically purify the protein, which he did in a very elegant way, starting with squid optic lobes. And then he wonderfully characterized the motor after he had purified it. By the time I decided to tackle this problem, techniques in DNA and RNA sequencing were very well established. And of course, you know about the human genome, for instance, which is the sequence of a human's entire genomic DNA, all of the chromosomes of a human being. And that work was being done by Craig Venter and others at the NIH and around the world. And I wanted to sequence not the DNA, not the genomic part, and I didn't want to biochemically purify the protein necessarily. I wanted to get after RNAs that would make kinesins, different kinesins. Were there more than a single kinesin in the squid giant axon? That was my question. And it had been shown in many other organisms that there's not a single gene for kinesin, but a family of genes. And in humans, there are about 45 different kinesin molecules, different genes that make different motor proteins responsible for doing all sorts of work in the body. For my sequence project, I took a squid like this, and I cut the mantle down the center. And I've said this before, it's like a hollow ice cream cone. And if you cut one edge of it, you can then unfold the ice cream cone. So it's something like this. And we said before that there's a neuron that comes down from the brain, and it synapses with a whole bunch of giant axons. There's not one giant axon in the squid, there's many, and there just happens to be one that's really long. And of course, these neurons bifurcate, and they innervate the muscle and control some of the swimming behavior of the organism. And there's not just one, they're symmetrical, so there's another set over here like this. And what I did is I was interested in what genes were expressed in the cell body of the giant axon. And all of these neurons come together in a structure called the stellate ganglia. So I took a pair of scissors and I just cut out this region here, which I knew would contain the DNA, the genomic DNA in the nucleus, of course, but then also it would contain the messenger RNA molecule. So I wanted to look at messenger RNA. So once I had a test tube full of about 100 stellate ganglia, I dissected out. I used 50 squid and dissected out 2 stellate ganglia from each squid. And I homogenized it, that is, I ground it up biochemically, and I purified the messenger RNA molecules. It's pretty simple, and you can buy a kit that there's a column that binds to the poly A tail because it has stretches of poly T's, and you can purify the total messenger RNA. So these are all of the genes that are expressed in the cell body of the giant axon. So whatever's being made in those particular cells. We've said this many times, but you can convert the RNA molecules into double-stranded DNA by binding T's to the A's and using reverse transcriptase, which makes a complementary strand. So if this is a G, this is a C, and so on. And then we chew up this top strand, this top RNA strand, and we add DNA polymerase, and it makes a complement to the complement, and now builds a double-stranded DNA molecule that's identical. The top strand is identical to the original RNA molecule, except all of the U's have been replaced by T's. So we can do that in a reaction that gives you all of the different RNAs. So it's not just RNAs for kinesin, it's RNAs for every single protein, every single gene that's being expressed in that particular cell type. So you could have, a messenger RNA that is for actin, for instance. So this one might be actin. Then you could have one that, is, yes, kinesin. We would predict that kinesin would be in the squid giant axon. Well, we know it's in the squid giant axon because it's in the S1. You could have an RNA for tubulin, which make up microtubules, and so on. So we know that there are about 20,000 genes in humans. We don't know how many that there are in squid, but at any rate, any cell type could be expressing as many as 20,000 genes, 20,000 different genes. So there's 20,000 different RNA molecules, and now they've been converted into double-stranded DNA. And now we can take each of those strands and insert them into a plasmid, just like we talked about before. So you could put this one in here. This is the actin, and then you could put another one, the kinesin. It's just random. Whatever RNAs were present were converted into double-stranded DNA that they call copy DNA, cDNA, and they all get inserted individually into a plasmid, and then each plasmid gets inserted into a bacteria. And then the bacteria are placed on a petri dish with some growth media, and the individual bacterial cells, each of which contains a different gene, like actin or kinesin or tubulin or whatever, they divide. And when they divide, they replicate the plasmid. So if you start out with one bacteria with one copy of the plasmid that has the tubulin gene, the tubulin double-stranded RNA, cDNA of tubulin, it has a copy here, and this one has actin, and so on. But anyway, each one of these bacteria would have its own unique plasmid, and each plasmid would have a different cDNA molecule inside the plasmid. And if you leave this overnight, these individual bacteria replicate. So 1, then 2, then 4, then 8, then 16, 32, 64, 128, and so on. So they very rapidly form a colony, and an individual bacteria on the plate is invisible to your eye, but overnight you can get a culture, they call these colonies, and each bacteria in the colony now has a copy of the plasmid that the original bacteria was given. They call this a cDNA library. So you have all of these different bacterial colonies. Then what they do, or what I hired a company, actually the NIH did this for me, the group that sequenced the human genome at the NIH did this part of the work for me. They have a robot, and the robot takes a toothpick and it sticks it into the colony of bacteria, and then it puts it into a test tube with growth media. So now you have this bacteria culture in growth media, then you have another one in the second tube, and so on. And I raised $120,000, and for that they selected 22,000 bacterial colonies. So they were going to grow up 22,000 colonies. So if you put a little bit of this bacteria in here and you shake this test tube overnight, the number of bacteria really rapidly increase. And so now you've gone from a single bacteria on the plate to many bacteria overnight to millions of bacteria after this culture is shaken for another 24-hour period. Then they spin down the bacteria, so they end up with a pellet of bacteria like this, and they throw the supernatant away, and then they biochemically purify the plasmid back out of the bacteria. So now they have 22,000 test tubes that have millions of copies of each of the plasmids. So, this test tube has this gene, and the next test tube has the next gene, and each one represents an RNA molecule that was isolated from the squid stellate ganglia that contains the cell bodies of the squid giant axon. And then they sequenced each insert. They need many, many copies to do the sequencing reaction. That's why you go through this process of putting it in bacteria and trying to amplify the number of bacteria, because every time the bacteria divides, it makes more copies of the plasmid, and you need lots of copies of the plasmid in order to sequence the original RNA molecule. Or in this case, they're sequencing the cDNA molecule. But if you know the cDNA molecule, you know the RNA molecule sequence, because you just remove the T's and stick in use there, and you've got the original RNA molecule again. So when they do the sequence, of course, that's just the order of the nucleotides on one of the two strands. So in the end, the sequence group gives me a document, a Word document, and it says sequence number 1. And it says, TAG, the whole sequence. And then it says sequence 2, like that. Okay? And I have the sequences for 22,000 RNA molecules. And then you have to analyze the data. So you have to take each one of these sequences and you go to PubMed, where we have the NCBI database of all gene sequences that are known, or, well, maybe not all that are known, but most people end up putting their sequences into that database. So the majority of sequences are in that database. And you tell the computer program, hey, what does this sequence look like? And it tries to match this sequence with all the nucleotide sequences in the database, and it finds a match, if it can. If it matches something, it will pop up and it says, hey, this thing is 95% similar to actin in human. So that would suggest that this is the squid version of actin. And then the next one might be, 97% similar to tubulin in cow. So it's probably the squid version of tubulin, and so on. And I did that for 22,000 sequences. I mean, I automated the process, but it still took weeks in order to analyze the data. And now you have, if you ask the computer program to limit the output to something like 4 pages long, so it's 4 times 22,000, so I end up with an 88,000 page document. So it's a really long document. But now if I want to search for a particular protein, like a kinesin, I just do a find command in Microsoft Word, and I type in kinesin. And the program scrolls down through the 88,000 page document looking for the word kinesin. And if it finds it, it stops. And you can see at that point that one of the sequences from squid that was put into the database matches to a kinesin sequence from some other organism. So you might find a kinesin that is, 99% similar to kinesin 1 in rats. And so you know that, well, that's a kinesin. It's probably a kinesin. I mean, it must be a kinesin. And in that way, I could continue to search through the document until I found all of the matches of genes I put in to the computer program that match to different kinesin molecules in the database. And if the kinesin molecules that I put in the database, now have been identified as kinesins, are not identical to one another, that suggests that they are from a different kinesin gene. So in that way, I could identify about 6 kinesins in squid, including the RNA molecule that encodes Ron's kinesin, but I found an additional 5 kinesins that hadn't been previously known in squid. Okay, a couple of notes before we move on. I said that we didn't know how many genes are expressed in the squid genome. In order to do that, we have to sequence the squid genome, and then we have to carefully analyze it to try to find the genes. We haven't done that yet. In humans, we think there are about 20,000 genes. When I say that I sequenced 22,000 different RNA molecules, I don't mean different genes, because some genes in the cell body of the giant axon might generate a huge number of RNA molecules. RNAs from genes like actin and tubulin, which make up the cytoskeleton, are expressed in really high amounts. So you can have many, many copies of actin mRNAs and tubulin mRNAs, but other genes might crank out just a few, like the kinesins, for instance, or the gene can be shut off altogether, so there are no RNA molecules for certain genes in certain cell types. So I sequenced 22,000 different random RNAs from the cell, and some of those are duplicates, actin over and over and over again. And then once in a while, you see, oh, this one's a kinesin, or it's tubulin over and over and over again. And every now and then, you find a dynein or a myosin, something like that. Now, it's highly unlikely that I've identified all of the kinesins that are expressed in the squid giant axon, or certainly in the squid itself, the squid overall. But I had identified 5 new kinesins, and adding that to Ron's, we had 6 kinesins that we could now study. So now I end up with 6 different kinesin molecules. Identified from the RNA, not the protein. Like this. 1, 2, 3, 4, 5, 6. And I had the double-stranded cDNA for each of these molecules. And of course, their sequence is a little different from one another. But the part of the molecule that's highly conserved are the heads. And so I wanted to make antibodies against each one of the kinesins, and the sequence is more divergent in the tail region. So I wanted to make antibodies that wouldn't recognize all of the different kinesins, because if this region of the molecule is conserved, the heads, they're all basically identical in nucleotide sequence. That means that they would be identical in amino acid sequence. And if you use the amino acid sequence to generate the antibody that was built against one gene might recognize all of the kinesins, because it's raised against a part of the kinesin that's the same in all of the different 6 molecules. So I picked a region for each one in the tail that was more divergent, and I could compare the sequences because I had all of them. I could compare them to one another and pick regions of the tail that would provide a unique antibody just to that species. So, I would, down here is where the tail would be encoded. If the start site is up here, then this part would encode the head, and then as you move closer to the stop codon for that messenger RNA, you would find the tail domain. So I PCR amplified a little stretch of the cDNA that encoded the tail of each of the 6 different molecules, including this one here, which was Ron's. And I, again, ligated this into a plasmid like this, each of them. So now I have 6 different plasmids, each of which contains a different tail from each of the 6 different kinesins that I identified. And then I put that into bacteria. And then, I mentioned this before, you can turn this gene on and you will make protein that is the tails now of this particular kinesin. And similarly, the tails of this particular kinesin. And then I can use that his tag polyhistidine that I mentioned, which are amino acids. So you just add a string artificially to the end over here. And if you get about 6, they will bind to a nickel-based column. And you can grind the bacteria up and run the sample over the column. And the tails now will bind to the column. And you can wash everything through and then elute off the protein by changing the pH. And it drops off. And so you now can have purified kinesin tail from kinesin 1 to kinesin 6, and you can inject each of these tails into a different host, like 6 different rabbits, for instance, and each of the rabbits will produce antibodies against the different types of kinesins. So I did that. So now I have 6 antibodies against 6 different squid kinesins. In this paper, I talk about 2 kinesins. I talk about kinesin 1 and a kinesin type 3. The paper is about this molecule, squid kinesin 3.